All right. So, I think this is working. All right. So let's go ahead and get started. As I alluded to yesterday when we started this little traveling notes thing, and I didn't say much. I just kind of sent you on your way. But essentially, what we're looking at here is how we go from these letters of T's and A's and G's and C's, how we go from this code to an actual real thing, which is a trait. Okay? And the idea is that these are instructions, okay? This code is an instruction set on how to build a protein. Because that's what all your traits are. How tall you are, how long your bones are, your skin tone, your hair color, all of those things that make you again, your brain cells, everything is built out of proteins. So what you're really looking at here, all of these letters are really instructions on how to build a protein. And that's why we've said all along, hey, the order of the nitrogen bases gives you your traits. It's because it's like an instruction booklet. The order of the instructions, this protein. The order of the instructions, this protein. The order of the instructions, this protein, and so on and so on and so on. Until you are, built uniquely you. Okay, so what we're going to look at is kind of the steps to take on how we go from this to that protein. And there's two processes involved. The first one is something called transcription. Okay, and that's what we're going to look at first. The second one then is called translation, and we'll look at that one here in a minute. So, with transcription, as we just kind of talked about, the information is on DNA, right? But there's a little bit of a problem. I'm going to go ahead and turn the light. That's a little dark. There you go. See, I'll see a little bit better. Yeah. Okay. There's a problem though, right? Because let's think back to when we talk about cells. Proteins, we said, were always built at the what? It starts with an R. The ribosome, yeah. So the ribosome is where proteins are built. So if this whole thing, this is all about building proteins, well, we've got to get DNA's message. We've got to get these instructions to where proteins are built. But what's the issue there? Does DNA, have we ever talked about DNA leaving the nucleus? No. DNA doesn't leave the nucleus. It's way too big. So the instructions on how to build the protein can't get to the site to build the protein. So there's got to be some kind of intermediary. There's got to be some kind of go-between that takes the instructions of DNA to where the protein's actually going to be built. Okay? And that something is RNA. RNA is the messenger boy, per se, to take DNA's instructions to where the protein's going to be built, to where that the ribosome then can read it and put together the correct protein. So think of it like this. You know me, I like to use goofy examples. I think they help you remember things. So think of it like this. DNA is an architect that has a disease called agoraphobia. Anybody ever heard of agoraphobia?
Yes. No.
You know what it means?
You're afraid of the spaces.
Yeah. You're afraid to go outdoors, right? You have to stay in enclosed places, basically. So DNA is this architect, a brilliant architect, that has all these brilliant ideas, but DNA can't leave, the architect can't leave its office. He's too scared to go outside.
So he sends it on email.
So there's no technology like that in DNA's world, unfortunately. So DNA has this worker, this messenger that works for him or her, and comes into the office, takes the instructions, writes them down, and then goes to the job site of where the building's going to be built. Okay? Well, that person that's taking the architect's instructions is like messenger RNA. The job site then would be the ribosome. Okay? And then the finished house or building or beautiful structure, ta-da, would be your protein. Okay? And all the pieces that are put together to build that protein would be the amino acids, because that's what proteins are built out of. Okay? So that all that I just told you is pretty much the whole process of transcription and translation in a nutshell. And now we'll just kind of look at the specifics on how it's done. You're going to notice, especially with transcription, you're going to notice it's very similar to DNA replication. Remember how DNA is split apart, free-floating nucleotides attached, we use the base pairing rules, A's and T's, G's and C's, and then ta-da, you get your two identical strands of DNA. This transcription is very similar, except we're using RNA free-floating nucleotides instead of DNA free-floating nucleotides. Because what's the goal? The goal is to transcribe DNA's message into a piece of RNA, messenger boy, that can leave the nucleus and go to the job site. Ribosome. Okay? So let's see how that actually works. So I'm going to, if you are following along, I pretty much just said the first like 2 or 3 slides. So we already talked about this. RNAs, where RNA comes in, DNA's information is copied into RNA. RNA goes to the ribosome. And let's look real quick, though, before we dive into the steps, a few differences between RNA and DNA. So one, DNA is double-stranded, and it's really large. So that's you know, I know my goofy example, the whole architect that's scared to leave his office. The real reason DNA doesn't just do it itself, it's too big. DNA is really large, and it can't leave the nucleus. It can't go out. And so it uses RNA, which is a smaller molecule because it's only single-stranded. Okay? And so that's the biggest difference. DNA is double-stranded, RNA is single-stranded. The type of sugar they use, okay, deoxyribose is for DNA, just regular ribose is for RNA. And then the biggest difference that you guys need to know about is right here. And I know a lot of you noticed that, and a lot of you talked about it yesterday, but uracil is used instead of thymine. So when we're doing our base pairing rules, when we're going from a set of DNA to a set of RNA, you've got to remember RNA doesn't have any T's. So if there's an A in the DNA strand, it's going to match up with a U for the RNA strand. Okay? And we'll practice that a couple times here in a minute. It won't be a big deal. Okay? All right, so let's look at the process. So step 1, just like replication, DNA has to separate. So it unwinds, it unzips, an enzyme breaks apart those hydrogen bonds, and then you have these 2 separate strands of DNA, just like so. The second step then, is again very similar. These free-floating nucleotides attached to the appropriate side, except it's not DNA nucleotides, it's RNA nucleotides, okay? So that would be like, ta-da. So let's just real quick practice that up here. So let's say if this is our DNA, and we'll just do a short strand, we'll go TAGCA, okay? So there's our little strand of DNA. We're going to transcribe that into a piece of RNA. What would be our base pairing rules here? T's go with A. Does RNA have T's?
Yes.
RNA doesn't have T's. RNA has U's instead of T. So A matches with U.
C, G,
U. Perfect. So ta-da! We just did transcription. This RNA then would go out to the ribosome, and the ribosome would read this code, put together the appropriate amino acids, and ta-da! You make your protein. Okay? Now, important, like, test question stuff. Where does transcription happen? Where does it occur?
In the cell.
In the cell, but specifically in the cell.
The
Yeah,
nucleus.
in the nucleus. Because does DNA ever leave the nucleus?
No.
No. So if DNA is involved in the process, Ta-da! It happens in the nucleus. Okay, you got a question?
Yeah, so is it just the old ones that go? It doesn't bring like the whole strand?
Oh, it will. It's just showing you that it's a process. Yeah, it would. It would keep copying there and there and there. Now, there are certain points that it would kind of read a certain code or a certain set of letters and know to stop. So it doesn't just like the whole DNA strand. Again, it's making specific proteins, right? Which we know are controlled by specific genes, which we know are only portions of our DNA. So it's going to read just the portion that it needs to read. Okay, good question. All right, so that's really about it. So as far as transcription is concerned, so DNA gives its message to RNA, and then RNA will leave, DNA zips back up, and that's pretty much it for transcription. Okay, so RNA separates, RNA goes to the ribosome to help make the protein. There's that single-stranded piece of RNA. And then the very last step, DNA is going to zip back and just stay in the nucleus. Bam. Done. Okay? So that's transcription. That's the first part. Again, our scared architect in his office, messenger boy comes in, writes down the blueprints, copies the blueprints, takes the blueprints out to the job site. Bam. That's transcription. Okay? Now, the second step. Okay, the second step is something completion. Okay? And let's see here. Oh, there we go. All right, so translation now is, again, kind of what it sounds like. So now we have this code, we have this blueprint. So now the ribosome has to translate this into the language of amino acids. It has to read this and say, okay, this is going to code for this amino acid, this amino acid, and this amino acid. So it's basically like the messenger boy going to the job site, lays out the blueprints, and then starts saying, hey, all right, 4 2x4s. We need them right here. Let's go. Bring them over. Bam. Brings them over. The 2x4s would be kind of like the amino acid. Then says, all right, now we need, 6 4x4s. Bring them right here. Let's go. Put them right here. 4x4s are the next amino acid. Then the next one. Then the next one. Then the next one. And again, you put it all together to build the house/protein, right? Okay. But again, how does it work? Well, that's what translation's all about. So let's look at how this actually occurs. So again, pretty much what I just told you, the decoding of this message is it happens in the ribosome, and that's what translation is. Who watched, a lot of you probably watched The Christmas Story over the break, right? That You'll Shoot Your Eye Out movie. Yeah, it's on like 24 hours Christmas Day. Shake your heads like you, yeah.
Your eye
Right,
out.
yeah,
The
whole
Rider?
with the little boy, Red Rider. You never seen that movie?
Freeze.
Reese. Oh my gosh. Anyway, so if you've seen that movie, you know that part where he goes, remember he gets he's waiting, like every day he checks the mail, he's waiting for that decoder ring or whatever, okay? And he goes up to the bathroom and he's like decoding the message, and all these different little numbers are a different letter, and he writes out the thing, and he's so, thinks it's going to be some big message and it's just a commercial. Ta-da! That's what this is. We're decoding a message, okay? And we have the help of a secret decoding chart. So if you will turn your book, everybody should have at least one book on your table, and I can get you another one if y'all need one. Page 234. Page 234. Look on page 234. Alright. So, page 234 has our secret decoding chart to kind of look at how we're actually going to get the amino acid in the right spot. Okay? Now, before we dive into this, let's look at one more thing here. There's a couple, we've already talked about messenger RNA. Messenger RNA, like it sounds, okay, is the messenger boy. It takes DNA's message out of the nucleus, to the ribosome. That's messenger RNA. There's a couple other types of RNA that are important as well. Transfer RNA. Transfer RNA, again, does what it sounds. It's the thing that will transfer the amino acid to the right spot, okay, on our job site. So again, RNA, messenger RNA gets DNA's message, okay, the architect's message, goes to the job site, okay, lays it out, starts calling for the appropriate materials. Hey, the 2x4s go right here, the 4x4s right here, and so on and so on. The people, the workers that bring the 2x4s over are transfer RNA. They're bringing the appropriate material to the right spot. Okay, the material again, what are they carrying? Amino acids. Okay, so transfer RNA are the workers that are bringing the material over. Just to kind of put it in those goofy terms. And we're going to do a little modeling session here in a minute that will kind of bring all this together. All right, now I'm going to turn the screen off because we're going to go to the board and we're going to draw some things out together. So look at wherever it says codon and anticodon on your single sheet. Okay, that's kind of where we're going to leave off, and then we will go from there. Okay, so all right. So here we go. So let's start from the beginning. All right. So here's our, and you can, if there's blank, there should be a whole blank side on the back of your your translation notes. If you want to follow along and draw this, it'll be a nice little reference. If you think you can just watch it, that's whatever helps you. So here's our line of DNA. Okay. So we'll go A, G, T. C, C, G, A, T, C, or no, A, T, A, sorry, and then G, C, C. Okay? So now what's our first step? Remember, we got to transcribe the DNA to RNA. Alright? So now, here comes the messenger RNA. It's going to copy down the code. Remember, we use our base pairing rules. So A's go with U. Bam. All right, perfect. So now we've just done, what's this process called that we just did?
Transcription.
Transcription, good. And where does it occur?
In the nucleus.
So perfect. So, ta-da, that's transcription. Okay, and it happened in the nucleus. All right, so now messenger RNA is now going to take that message, okay, to the ribosome to build the protein, right? But the ribosome doesn't just read this whole line; it chunks it up. It chunks it up in sets of 3 bases. So now I want you to block off 3 bases. So 3 at a time, 3 at a time, 3 at a time, just like that.
These in there.
Hey, they're good there right now.
Okay.
Thanks, Molly. So referring kind of back to your notes now, these 3 base pairs of messenger RNA is called a codon. So that's a codon, that's a codon, that's a codon, that's a codon. So every 3 bases of messenger RNA is called a codon. The codon then is what we use to find out and to translate the amino acid to the right spot. So now this is where page 234 comes in very handy. So if you look at that, what's called a codon chart, it's super easy to use. You go left, top, right. So if you start on the left with your first base, so U, find the U, it should be the brown one up top, okay? And then go to the top and find C. So now that should, if you follow it like a little graph, U and then the top C, it should take you to a block And then on the right side, find the A and follow that one over. And it will bring you to the specific amino acid called
Stop.
serine.
Never mind. Siri.
Serine. Perfect. So the amino acid that goes with this codon is called
Siri.
serine. Okay, so bam. Serine's placed right there. Let's look at the next one. GGC. Remember, left, top, right. GGC. What is it? Glycine. Is everybody seeing it? Give me a thumbs up if you found glycine. GGC. Left, top, right. Okay? What about UAU? Tyrosine? Left, top, right. UAU. Tyrosine, everybody see it?
I get what you're doing now.
And the last one, CGG.
Arginine.
So again, left, top, right, CGG, arginine.
I'm disgusted.
And ta-da! What did we, now again, think in terms of the processes, right? Transcription, translation. What did we just do? Transcription or translation? Translation. We translated these codons into amino acids. Okay? So that would be translation. And where did we do this? At the ribosome.
After our little messenger.
Right. So again, back to our goofy example. Scared architect is DNA. Gives his message to the messenger boy. Goes to the job site. Okay? Ta-da, that was transcription. Now, translation is going to read part of the blueprints. We're reading part of the blueprints to see what materials we need at what spot. 2x4s here, 4x4s there.
Nails.
Nails here, screws over here. And ta-da, now we put it all together to build our house or whatever. Okay? So now the missing piece that hopefully some of you are catching is, well, wait a minute. I thought Strout said something about tRNA. And if I look at my notes, there's something about these anticodons. Like, what the heck's all that? All right? Well, remember we said there's got to be a worker that brings the 2x4 over, right? It doesn't just magically appear.
Yeah.
Well, the worker that's bringing serine over is tRNA.
So does he like go outside of the cell to go get it?
It's all inside the cell.
Is it a boy?
It would all be in the cytoplasm floating around. Yeah. And really, the amino acids, it's from our food. A lot of amino acids, some of them can be made like by our bodies, but some of them we have to actually consume. Okay? So look, what tRNA does, again, just like it sounds, transfer RNA, it's going to transfer the appropriate amino acid to the right spot. So look, but again, how does it know where to take it? Well, if these are codons, then tRNA has anticodons, which would be the opposite, right? So UCA, if that's the codon, the anticodon would be AGU. And that brings serine over. What's the anticodon for GGC?
CCG.
CCG, just the opposite. That brings glycine over. Okay? And again, the set of 3 mRNA is called a codon. The set of 3 tRNA, anticodon. Alright? So now I need, well, let me shut off that's about it for the recording.